So yeah, so Edward.
Yeah. All right. So second lecture. So it's going to be a bit of the reverse from the previous one. So I'm going to start with some more pedagogical background, but it's going to be a bit shorter. This time the bulk of the lecture will be more about sort of ongoing work as well as published. So what I want to talk about, so last time I talked mainly about sort of collective migration and ordering through sort of self-generated gradients, and giving you backgrounds from this mathematical biology literature. There's of course the counterpart in the physics literature, which has been very interested from the start at looking at how you get polar order in actively moving objects. And I'm sure many of you have heard this before. I think Christian mentioned it. This maybe sort of the paper that sort of could be said to have founded the field of active matter, so 30 years ago, which is the Vicsek model, right, which has been aiming to look at this kind of beautiful, to explain this kind of beautiful pictures where you have like all of these birds, or in general many animals, that each move in a directional manner and manage somehow to synchronize to all sort of go in the same direction, right? So mesmerizing things. And these are at the macroscopic scale, but there's also quite a few examples of things like this that happens at the microscopic or mesoscopic scale. Microscopic scale, for instance, there's this really nice work from Andreas Bausch from 15 years ago, where if you look at sort of self-propelled actin and motors, you end up with sort of self-propulsion of your particle at the inside, which gives rise to this sort of ordered states where everything moves at the same time. That's microscopic. That's macroscopic. Actually, what I'll talk about, as you might have guessed, is more the mesoscopic scale and how this applies to collective cell migration. So in a way, this is a very different approach, but it is complementary to what I talked before. What I talked before was we were not actually considering any explicit cell-cell interactions or polarity interactions. The interactions were only happening through a diffusible field, but through this you could get an actual direction, right? You could go sort of left or right. Here it's very different. This model is not going to specify which direction do you go, but it's going to tell you why does everyone polarize in one direction and picking that one at random. And of course, I won't talk too much about that, but it's easy to imagine that right now we can start blending these kind of models where these kind of sort of polarity interactions can give rise to long-range order in any direction, which then you can modulate in the preferred direction that you want based on relatively small gradients in morphogenesis, right? So these are not opposite ideas. They're very complementary. They're very complementary, in fact. Okay, so I think that Christina mentioned frocking. I'll still, but relatively briefly, I'll still go very briefly in the history and the ideas around frocking. So as I said, this is sort of the Vicsek model from '95, which has been hugely influential to the field of active matter. And it's a really simple model. It's something which is essentially where there's essentially no mechanical interactions. So you consider sort of cells or particles or whatever, boids, as polar particles. So they have a spin and they move in the direction of the spin with velocity v0. That's it. They don't feel anyone. They don't feel sort of there's no repulsive interactions, no attractive interactions, absolutely nothing. But the important thing is that when they get close to other neighbors, they change their direction and they align to their neighbors in some sort of small radius r0. So if you write the equations for that, basically, if you take the position of a given spin I, it's going to move. It's going to move in the direction of its velocity vi, and then the angle. So let's write it in 2D because it's easier. The angle of that velocity vector is basically at every time point going to be an average of the j's, which are essentially the neighbor list of I, plus some noise because they're not good, because they're not perfect at aligning to neighbors, right? Okay. So one point which is important here is that, and it's going to be important in the lecture, again, this is a completely non-mechanical model. Which means that if you think about the talk of Julia and the language that she mentions, polarity and velocities are always aligned, right? Sort of, if you can think about the spin as the polarity of the cell that they migrate in, but because they don't have any mechanical interactions, no one pushing you in that direction, your polarity and your velocities are basically the same thing, right? And that's going to be important because if you have mechanical interactions, that doesn't have to be the case because you can want to move in this direction because that's your polarity, but you can be stopped by everyone else if there's sort of obstacles along the way, right? So that's something relatively important to mention here. So why you might ask, okay, why is this model chosen? Why is this model chosen? What's quite beautiful from a physics perspective is that this looks very similar to the XY model, right? This is almost exactly what you would have in an XY model in the sense that you have a spin that can rotate, that has a continuous symmetry and can sort of rotate at any point. It's in 2D. And then it aligns to your neighbors. And basically, the only difference from the XY model in this is that you can move, right? You move in the direction of your spin. And so it's kind of nice because it's in a way the simplest non-equilibrium sort of variation that you can do out of the most classical, sort of probably the most classical model of statistical physics you can imagine. So this is just to go back, right? If you put your velocity v0, this is the XY, this is the classical XY model. And the reason why people got so excited into this is, okay, this is 3D, so it's not a very good example. But if you run these simulations in 2D, you have long range order, which means that if you have sufficiently low noise or sufficiently high density, everyone is going to move in one direction. Everyone is going to align. Which in the language of the XY model is a non-zero magnetization, and that's forbidden by equilibrium physics. Because in 2D, I mean, I'm not going to enter too much into the detail, but it's a continuous symmetry, it's 2D, so it's a fundamental theorem of equilibrium statistical physics that you can't break symmetry in 2D. So it means that something is happening, and it means that the fact that you're moving in the direction of your spin fundamentally changes the physics of the problem. And again, I'm not going to enter too much into the detail of this, but I'm just going to give you a very simple mean field argument. The key thing in here is that because you move, essentially because you move, you can encounter more neighbors, right? If you think about for instance, an ordered phase where everyone moves in one direction, in the classical XY model, if you have a small deviation, you're going to be sort of rectified by your neighbors that are around you. But in this model, not only that, if your spin deviates a little bit from the neighbors, you're going to start bumping into them, right? Because suddenly you're going to have a relative velocity compared to your neighbors. You're going to bump into that, and that's an additional stabilizing term. And so there's something fundamentally new that shows up because of your I mean, it's short range interaction. I mean, the point is that the R0 is small, and so it's short range interactions. And so, I mean, you don't change the universality of, I mean, you don't change the universality of your XY model by just having nearest neighbor or changing a little. As long as you have short range interactions, things don't matter so much, right? That's the point. So it's yeah. It's also an off-lattice version of it. Yes, you're right. Yeah. Yeah, no, absolutely. Yeah, no, absolutely, yes. No, yeah. It's more general. You need a continuous symmetry. I mean, it's more about the continuous symmetry of your spin. So that's all right. So the question is why do you, so the classical phase diagram of this kind of Vicsek model essentially is if you put the noise on this axis and your density on this axis, if you're dense enough or low enough, if you're dense enough, you're going to get an ordered phase, and if you're noisy enough, you're going to get a disordered phase, right? And so this was one of the closed exercise I gave. I can actually, I think, give the solution. You can rationalize this in a very, very simple manner through a very simple mean field argument, which is good for low density. The point, right, is if you think about things like, if you think about birds, for instance, that move, the point is, let's say that you imagine that you have two clusters and they move in different directions. The question is, do they tend to align with each other or do they tend to lose directionality? And the key idea behind this is that your collision time, what allows you to repolarize in the same direction is that you have essentially a collision, that you get close to this. And that's obviously density dependent, right? The more dense you are, the more you're going to collide and have chance to reorient. But the more noise you have, the more your orientational order is going to be lost, right? And so the key thing is that the distance of, I mean, if you look at the mean sort of at the typical length scale, you can do this either as a function of length scales or time scales of the problem. The key thing is that the characteristic distance between particles scales as 1 over rho 0, right? And if you think about the time scale at which so essentially your particles undergo rotational diffusion with noise eta, right? So they lose the time it takes to lose your directionality is 1 over eta squared. And so if you convert this into the length scale, it's how much distance will you have covered by that time. So it's V0 over eta squared. And so the very simple mean field thing you can say is what matters is that if I collide more frequently than I have time to lose my directional order, my direction, I'm going to order, and if not, I'm going to disorder. So what essentially this says is that if you equalize these two things, you can have ordering if your eta, if your noise is lower than square root of V0 over rho 0, which gives you sort of this sort of square root sort of relationship, which is sort of a bit sort of kind of nice with what you see here. So it's nice, right? It makes a lot of sense. You get ordering for low noise, and you get ordering for low noise or for high densities. And you can also see something which is quite nice, which is that if you put V0 to 0, right, if you put the system to the equilibrium limit, there's no transition anymore, right? Because the transition happens at any sort of amount of noise destroys your order. So it's kind of nice, right? It's a very simple way to understand the physics of this. And of course, it's much more complicated. There's a middle phase with bands and everything. I don't really want to enter into this. But this is a really, really simple model where you get sort of fundamentally new physics as a function of having self-propelled velocities plus some very basic interactions. Okay. And some more slides. I'm not going to go into detail because it's the point. The thing I want to talk about is actually yeah. So this was in 2D actually. This was in 2D. Yeah. In 3D you can also get order, but it's less surprising because you can also get 3D get order in XY in the XY model. So in the generalized. Sorry, did I define it? Sorry, did I define it as a path? Sorry, that's a good point. Maybe I got the explanation. Sorry, I'm not so sure anymore now. I don't know if it's the definition of the density that is, or I don't know if it's about because or maybe it's because of your rotation. It's the rotational sort of thing. So it's the question of like how you meet in a sphere of R0. I have to rethink about this actually. That's a good point. I'm not totally sure. And it's mainly because you have to you're rotationally dividing, so you're rotationally moving around. So it's whether you meet in a whether everyone has a domain. Maybe I think it's probably that everyone has a domain, right? That you want to meet before you meet in a domain. That's something like this, I would say. So this actually is something that I want to connect to what Julia mentioned. Is I think this has also so I've shown you example in a macroscopic and a mesoscopic scale, but this has also sort of had a lot of influence on how people think about cells. It is actually exactly the same paper as Julia has shown you. This is quite a nice paper where they sort of knock out RAB5 and they see this sort of super global motion of this, which they interpret as a Flocking transition. And in general, this is a cell type which does sort of less spectacular. I mean, it doesn't have sort of global motion, but it clearly has sort of locally correlated motion, right? Where there's swirls, where your net motion is correlated. And so when looking at these things, it's quite tempting to think about sort of similar types of mechanisms occurring at the cellular level. And I should really say so. I'm in a next talk when I'm going to talk about what I'm going to talk about is describing the systems more as polar active matter. So in a way, I'm going to take the orthogonal view to what Julia was talking about, which is sort of describing on a kinematic. And I should really say that it's a very open problem, and we really don't know which one it is. And I think what I will find super interesting in the next sort of few years is to actually compare systematically these different things, because it's probably quite clear that they have some mixed symmetries and that a bit everything sort of happens in the system. And I find it, and it's very good. I mean, I think there's very interesting and convincing arguments for everything. And I think there will be like an important unifying sort of exercise in the next few years. And so when discussing about these things, what I wanted to say is that there's been a lot of theories done on that. So one is flocking. The other one, which I will briefly mention, is seeing this, for instance, as an active class. There's the question of nematic turbulence, and this has been looked at by quite a few people actually one of the first is in the audience, and I think one of the things that I want to talk about as part of it's a paper that just got published from our group, is I think it's important now to try to find ways to distinguish between, to sort of systematically look at different couplings and try to distinguish what would different couplings look like. Because of course there can be many sort of different ways, there can be many different sort of type of things that give rise to the same sort of collective behavior. And in particular, what I want to mention here, so this is actually a schematic from a really nice review from Ricard Allert and Xavier Trepa, which I think Julia also mentioned. It's by the way, it's a really nice review for physicists if you want to learn more about collective cell migration. It enumerates like many different models at different scales, etc., etc. And one of the key things I think that sort of this review makes clear is that what we don't really know is if we want to describe these things as sort of polar particles. For instance, when they heal a wound, it's clear that they have to be polar in some way. Is that we think we have some good equations for the velocity, right? Because it's probably viscoelastic. We have force balance plus viscoelasticity, so it cannot be completely crazy. So if you want to describe the velocity, I mean, we're not sure which one it is, but we have some ideas of Kenneth model, and Ulrich talk about a number. But one of the, I think, big issues is this polarity term, right? Like there's a lot of things that can couple. First of all, polarity is hard to manage in cells. That's been sort of, I think, an historic problem. So it's hard to get data on what the polarity looks like. And it can be, and just based on symmetry, there's quite a few things you can couple polarity to, right? And it's a bit unclear. So here, for instance, there's ideas of coupling polarity to velocity. There's contact inhibition of locomotion, which Julia talked about, which is polarity going away from cell density, basically. You can align polarity to your stress fibers, for instance, to your long axis, and then pick the velocity out of this. There's essentially many different ways you can do that. And
I mean, you used it yesterday too, because
yeah. It's probably the case, but I'm not sure if it's the case in every cell. I mean, I'm not sure if it's the case in every cell type. I don't know how. I mean, it's probably a good readout on average, but is it, for instance, if you want to look at fluctuations, for instance, it might not. Yeah. 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 No, that makes sense. I'm not saying we have no idea how to read out polarity, but I think especially in the tissue, it's not very obvious often to get, especially if you want to look at things like long range order and where you need some precise readout of your fluctuations and things like this. It's a non. I mean, there's some possibility, but it's a non-trivial problem, I would say. Yeah, but in the tissue, it's hard. Usually, I mean, for single cells, it's very nice for a single cell, it's very nice to define lamellipodia. But usually in the tissue, they're cryptic, they're below, there's also actin pool. So it's actually, I mean, I'll briefly mention this in one of the next things, but it's not that easy to do even in tissues. You can use traction forces, but as Ulrich said, traction forces are quality, there's a long range elastic. I mean, there's a lot of, I mean, there's a resolution problem. So there are many possibilities, but they all have, I think, flaws, right? I think that's one of the issues with this. All right. Okay, so what I want to say in this part of the talk, basically, is I think one of the issues about distinguishing this is that very often we've looked at infinite systems. And the problem with infinite systems is that often it doesn't really matter what's the microscopic behavior, what's the microscopic you put. Everything converges on some universal sort of features, and it's very hard. I mean, it's what's beautiful about physics, but it's also a bit annoying if what you want to understand is actually the precise mechanism of regulation, right? And in principle, like essentially the two type of things you can write on polarity are things that are either sort of flocking and anti-flocking. You can make terms that tend to align cells with each other, and they're going to tend to all move together, or you can tend to anti-align them, and then they're going to form sort of macroscopic, force they're going to form macroscopic force dipoles where they're all going to be sort of anti-aligned, I mean, sort of large-scale anti-aligned with each other. And of course, many different cells could do this. Many different types of cells could have different types of motility. One thing which I find quite cool, and which I think is really suggestive and really guided us in this work, is that very surprisingly, it seems like the same cell type in several cases can actually have either flocking or anti-flocking alignments interactions as a function of boundary condition. So this is like a really nice work from Benoît Ladoux and Mej, where they actually plate MDCK cells on rings, essentially on periodic boundary conditions. And what you find in this case is that they start flocking. So they go around and around and around basically forever. So they're going to have sort of perfect long range order. And surprisingly, you can make these rings extremely large. You can make them into sort of complex geometries. It is sort of very robustly the emergence of a flocking-like mode where everyone sort of rotates and every cell has the same velocity. That's cool. But the really surprising thing is that if you put two MDCK cells with free boundary conditions, they don't at all sort of flock in the same direction. If anything, they anti-align basically. Sort of they do sort of something that looks more like contact inhibition of locomotion, where one goes in this direction and one goes in the other direction, and they pull on each other and they basically anti-align. And when you think about it, I mean, it's super weird, right? Because it says that exactly the same cell types have different interactions. The way we interpret this is that basically what it probably means is that even a given cell type has multiple of these interactions at the same time, but different ones get revealed by different boundary conditions. And so what I want to, and so this leads on some work we've done. So this is a collaboration. All the experiments have been done with the lab of Sylvain Gabriele, and all of the theory has been done by David Bruckner in my group, who's moving now to start his group in Basel in the Biocenter. Yeah. Yeah.
Is it that they start circulating when they have one continuously?
Absolutely. Exactly. That's exactly the case. Exactly. It's really they really need periodic boundary conditions, sort of N conference. That's exactly right. Yeah. Thanks a lot. Yeah. So 3D is more complicated because everything I'm going to rely on right now is that cells exert forces on a substrate. So they basically sort of, you break Galilean invariance because you can because you move by communicating momentum to the substrate, and that's nice. And that's nice. That's much simpler than the 3D case without matrix, where you have to consider that cells sort of exert force on each other, and then so everything I'm going to talk about is actually 2D because it's much simpler. And I think the problem in 3D, right? I don't know how you would flock because you need to. I mean, birds flock by sort of each moving, and they have air to exert resistance to. If you think about cells, they would need at least to be 3D, but with a substrate, with an ECM in between that they can pull on, right? Because otherwise they need to. I mean, forces have to be balanced. So there's a bit of a tricky thing on whether there's an ECM or not in this case, kind of systems. All right. Yeah. Oh, sorry. So that's a very good question. We want to look at this. We haven't looked at this. in the paper, they make pretty huge rings and even circuits like which are non-ring, and it's pretty huge. I don't know if they've calculated in this paper the time scale to order. Like how much does the ordering sort of depend on the length scale? I mean, on the size of the system. That's a good question. I think it's something we were thinking about looking, but we didn't. I think it's hundreds at least. It's a lot. It's quite a lot actually. It's a 1D train with at least hundreds of cells, so it's pretty large. And I don't know how this. It's a good question. I don't know. Yeah. No, so they describe this really. I mean, so the way they describe this, so they have a model in this paper. In this paper, they actually have a model, and effectively the model is flocking because they basically have something in which you switch your polarity based on the force that you're exerted, based on the force that's exerted on you. So they have, sorry? They do it essentially in 1D, yes. Yeah. All right. So what I want to, so I think the theme of this part of the talk will be one which is sort of more biological questions, sort of how are collective modes of migration affected by varying boundary conditions, and can we understand why different boundary conditions create completely different modes of migration? But there's one, I think, more general question for active matter is I think that what this project shows is that we can learn actually a lot about how active systems interact with each other by looking at how they react with boundaries at finite sizes. That's going to be sort of the sort of broader sort of point of this talk. So what's the system that Sylvain was looking at? Well, sort of again, it's very good because this was actually introduced by Ulrich in his talk. So these are actually fish keratinocytes. So these are these fish cells that Julie Theriault has. And what Ulrich showed is that it was single cells. So these are the cells that have like enormous lamellipodia, and they migrate super directionally. So if you take a single cell, it's going to migrate basically forever in one direction, sort of with relatively small dispersion. But it turns out that actually, sorry? Yeah. Okay. Yeah. You mean perfect circle? Perfect circles? No. Oh, really? Okay. Okay, I didn't know that. That's cool. I see. Okay. I see. Cool. I didn't know about that. Okay, that's interesting. But these cells, I mean, these cells are not isolated. They usually live in a tissue, right? They're scale, I think, scales things. And so they actually form adhesion. So this is actually a train of cell where these cells are adhered to each other. And so you see a few cells here, and they form really nice trains. Unfortunately, the movie is not running. That's very unfortunate. Essentially, but that movie is relatively boring. What happens in this movie is that essentially everyone moves at the same velocity. So these there's not really a leader. I mean, there's not really a leader or a follower. Every cell, the train moves at the speed of single cells. So they manage to all move together, and they form relatively strong adhesions. Okay. So that's the result, right? So that's the result quantified. If you compare trains of different numbers of cells compared to single cells, basically the speed of migration is not affected. That's quite different from what you would get from leaders or followers or something like this. So it seems like every cell manages to actively move and to contribute to the migration of these trains. Okay. So this in itself is a little bit not very interested. I mean, this is exactly what you would get, for instance, in this kind of flocking models, right? Where you sort of every cell has a polarity, and then they align to each other, so they go in the same direction, and they all move at the direction of single cells, right? But there was something which was a little bit more interesting, is that they can because sorry, I should have said they do these trains by making micro patterns. So basically, they print micro stripes where the cells can only adhere to these things. And so there's no boundaries here. So there's no like walls or something. They migrate on these channels because that's the only place where they can adhere. Sorry. Sorry? They always move in groups. So basically they have a pretty constant speed, and they just sort of trade through, and they're going to move, they move in that direction on the channel forever. No. So the thing which is a little bit more interesting is that they can also control now they can control not only the length of the trains but also their width. And so if you control that, what was a little bit surprising is that as you increase the more you increase the width, the more you slow down the trains overall, right? So you slow them down by having from 1 to 2, a bit more from 2 to 3, and a bit more from 3 to 6 or something. And that's a bit surprising because, again, Julia really nicely introduced this idea of contact inhibition of locomotion. The classical idea from biology is that the more contacts you have, I mean, when you have contacts, that slows you down, right? But here it says that having sort of lateral, perpendicular versus parallel contact is completely different, right? You don't care at all about having contacts in your direction of motion, but you care quite massively about having contacts in your direction perpendicular to motion, which is a little bit weird. And you might say, okay, initially we thought that's maybe because here they're not that efficient that they start swirling around. Like basically here they start not migrating sort of in one direction and they start sort of making unproductive motion by swirling around. But that's not the case. They essentially don't rearrange. They basically don't rearrange. It essentially they essentially move as a solid, but they're still much less efficient than before, which to us was cool. So this is what it looks like, and this is the problem of, I mean, this is the problem of measuring polarity, right? Is that in principle you could say that sort of this is the lamellipodium, so you can define polarity by sort of making this vector, but you can see that it's pretty messy inside our tissue, right? But okay, the point is that we have a phenomenon which at least to us is non-trivial. And even though these cells are relatively simple, and the question that we were asking is, can we understand which kind of cell-cell interactions gives rise to this non-trivial phenomena? So what David did essentially is, because we don't see any rearrangements, we can simplify things by having cell trains by essentially sort of elastically coupled active interacting particles. So basically, we're not going to change, we're going to make some hexagonal lattice, and we're not going to change cell-cell connectivity. So every cell is connected through a spring. So essentially, force balance is a bit boring. There's not much you can write, right? So this is just an overdamped equation where you have where you have springs, you have a polarity force. So this is the force that cells exert on the substrate in the direction of the polarity that moves them. There's some confinement, but that's just because you want them to stay on a stripe. So this is pretty boring. There's very little, I mean, there's not much else you can write. We wrote it at the linear level. You could write some nonlinear terms. I don't think it matters. So there's essentially nothing else you can write on this type. The interesting equation now is my polarity equation. And here we wrote something very flocking-like, which is that essentially what we know is that a single cell polarizes in any direction. And so the simplest thing to write is this kind of sort of flocking term, where essentially a cell picks a direction of polarity of unit, which is a unit vector in any direction. And there's some noise. And the key thing, and the key unknown basically is this thing, right? Is what are the interactions between cell-cell polarities? What is this term here? That's the one that we want to learn from the data. Okay. Everything is clear? Yeah, understand? No. All right. And so here again, there were a lot of proposals from the literature. And so because it's a relatively simple system, what David did is just to enumerate all of the possible couplings allowed by symmetry and test all of them basically. So again, it's not a very complicated model. So essentially, there's only 4 couplings which you can write out of biosymmetry because polarity is a vector, and there's essentially only 4 vectors you can make in this kind of system. One vector is velocity. That's the idea that you have that you align your polarity in your direction of the velocity. There's another, there's another flocking-like term where you align your polarity to the polarity of the neighbors, right? Polarity is another vector, so you can align polarity. I mean, both of these are flocking-like interactions. Again, in flocking, polarity and velocity is the same thing, right? So these are a little bit, these are a little bit sort of identical, but these are essentially flocking-like interactions. Then the other thing which has been proposed in the past is that polarity being coupled to gradients of stresses, right? And this, there's some evidence that, for instance, if you pull on a cell in one side, it develops a polarity in the other side. So that's again, gradients of stress is a vector, right? So you can write something where if you pull in this direction, the cells goes in the opposite direction. That's an anti-flocking term, right? Because it means that if a cell goes in this direction, the other one is going to tend to polarize in the reverse direction. So that's anti-flocking. And then there's this contact elevation of locomotion that Julia introduced, which is not exactly the same. It's a geometric term. It's basically the idea that your polarity aligns reverse to your neighbors, basically. So this is not linked to, this could look like it's a stress polarity coupling, but it doesn't require any stress. Even if your springs are at the rest level, you go your polarity in the direction opposite to where your springs are. And that's again an anti-flocking mechanism, right? Because it's going to tend to make a force dipole. Yeah.
The homogeneous term, E times 1 minus B squared. So that transition occurs as a function of density. So increase the density then.
Yeah, so here that's not really going to happen because the cells have a constant, because the trains are already formed, the density is constant basically. The springs between the cells, I mean, essentially, this is not going to be so relevant because the cells are connected through pretty stiff springs, and so therefore the density doesn't meaningfully change in these experiments. And the noise is going to be relatively small, which mirrors the fact that if you take a single cell, it takes forever to. I mean, I didn't know it made a ring, but it takes forever basically to repolarize. Okay, so now I think we enumerated all of the possible couplings to linear order allowed by symmetry. Yeah. I mean, these cells. I mean, it's very unfortunate I don't have the movie, but it's very unfortunate I don't have a movie. But these things, I mean, these things move forever, basically. So you have a single train of cells, and they basically are going to move in a directional manner forever. But it's really in the direction, it's always in the direction of lamellipodia. So there's a massive lamellipodia in front, and it always moves exactly in the direction of your lamellipodia. So it's, yeah. So the question is now, we have 4 types of interactions, and what David did is to systematically screen parameter space to ask whether we could understand our data, which is sort of global velocity independent on length, and critically dependent on width. And essentially, all models spectacularly fail at reproducing the data. And which is good, right? Because it means, I mean, initially it's disappointing, but I think it's good in a way because it tells you that maybe there's something non-trivial happening in there. And actually, there's a very good reason for why the models fail at recapitulating the data, because basically, if sort of flocking is dominant, if you put some flocking terms, you're always going to move it at constant speed, irrespective of whether you're wide or long, right? And if you put anti-flocking term, you're never going to have sort of net movements, right? You're always going to create a force dipole, irrespective of whether you're sort of wide or long. So then what we thought is, could it be that there's some mixtures of terms, basically? There's some flocking term that enables you to go in one direction, but there's some anti-flocking term that maybe sort of penalize, that sort of develop more for larger width, that sort of penalize your motion. So then what David did is to try out, again, to systematically screen all combinations of terms. So basically, he did all pairwise combination of interaction and screened all parameter values. Yeah. So basically, if you have some flocking-like interactions, here, this is the speed of the train as a function of either length or width. And basically, if you have flocking interactions, your the speed of the train is always constant. It never slows down with either length or speed. And what we can say in the data is that the wider trains slow down. And that's not something that you see in a flocking model. And similarly, if you do an anti-flocking model, as long as you have more than two cells, your speed always goes to is always very small, both in length and in width. So basically, nothing happens. So then we were, okay, can you do pairwise combinations of models? And can you sort of, with a pairwise combination of model, explain the data? And again, we tried all pairwise combination of models, and it again failed completely. So there's no pairwise combination. I mean, we're pretty sure that there's no pairwise combination of interactions that manages to reproduce the data. So then we went to the third order, and you can ask, okay, is there combinations of 3 interactions that reproduce the data? And then, yes, suddenly it worked. And it might look completely ad hoc, but I'm going to give you an explanation later for why it might be useful. I mean, we don't like, I mean, as physicists, we don't like three interactions is too much, right? But I'm going to sort of try to convince you afterwards that actually it might be useful for the system to have that. Yes.
And you're splitting panel with periodic boundary, filling the entire thing. So.
No, sorry. No, it's really a train. So it looks like this. It's a finite cluster, yes. It's a finite cluster where we change either the length or the width, and we simulate an infinite channel. Yes, basically. That, and which is also what we see in experiments. We never see any reversals.
Cells, individual cells have this as well?
Yeah. Yeah. Yeah.
The first bit is a velocity alignment already there, like x dot is alignment. So in some sense, velocity alignment.
I mean, it's not velocity alignment, that's force balance, right? So it's I mean, it has to be at least, yes, it has to be at least partially, but it doesn't have to be the case because of the confinement. For instance, if the polarity force are up, there's no velocity. So, right, and it will be relevant. Because confinement breaks the symmetry. I mean, basically, the point I'm going to get to is that confinement breaks the symmetry.
There is partial velocity alignment in every Vicsek.
Yeah. Yes, but I mean, yes, there's going to be some. I mean, there's going to be some alignment, but that's at the single cell level, right? That doesn't tell anything about neighbors. Yeah, but yeah. Okay. So what it turns out is that essentially we need sort of three combinations of interactions to explain the data. We need sort of velocity alignment, polarity alignment, and contact inhibition of locomotion. So the first two and the third. And if you do this in a relatively wide regime of parameters, you can explain very well your data where the length of the speed of the trains are length independent, and you can explain quantitatively their dependence on width. And so why is that what's going on? What's going on basically is that you can visualize it in a simulation where we've plotted the polarity. What happens is that you have both velocity alignments that tend to make you flock. So if you have a 1D channel, if you have essentially a train of one cell, everyone goes in this direction, which is good essentially. So essentially, so this you would get essentially just by velocity alignment. But then you also have contact inhibition of locomotion. And what contact inhibition of locomotion does is basically it polarizes you away from your neighbors. So as you get wider and wider, you get sort of these, this kind of sort of outward motion here, which are dissipating useful polarity, right? Because these ones are useless. And why do you need polarity alignment? The reason you need polarity alignment is that contact inhibition of locomotion only works for this layer here, the outward layer. If you don't have polarity alignment, it would only be this layer that would be outwards, and this one wouldn't know about it. But what polarity alignment does is to guarantee is to sort of propagate the mispolarity alignment here, which makes it gradual, which means that there's sort of which means that the whole thing is gradual. The more you widen, the more you widen, essentially, the more you make this kind of outward polarity, right? So that's the mechanism for which explains why as you increase the length of the cluster, you always have the same velocity. But as you increase the width of the cluster, you make polarity more and more unproductive by going outwards due to contact inhibition of locomotion. You can just sort of do simulations. So your simulations work quite well. So if you look at this, for instance, it's sort of very fast. It's very fast in 1D. But if you look at this one, for instance, it's relatively slow. It still moves because there's polarity alignment, but there's all of these unproductive arrows that propagate in the bulk that tend to go in the wrong direction, right? So the question is that true? So first of all, is that true? Is there all of this outwards polarity for wider clusters? The way to do it is to the signature of this is to look now at the stress in the train. If there's a lot of outward unproductive motion, you're going to strain your springs in the y direction, right? So the more you widen the cluster, the more you're going to create sort of tensile stress in the Y direction. And that's good because that's something you can test with traction force microscopy. So it's good. This was introduced before. We worked with the lab of Xavier Trepa on this. And basically, what we show is that for thin trains, essentially, there's very little there's very little sort of component of the stress in this direction. And as you increase the width of the clusters, there's more and more stress in the unproductive direction compared to the productive direction. And this is the model parameterized from before and predicts this evolution quite well. So that's nice. What I want to say is that now what I want to go back to is, you might say, okay, this is a bit ugly to have 3 interactions. Like, first of all, it's a bit ugly. Why do you need 3 interactions? It's not very elegant. But also, it seems extremely wasteful, right? Why the hell would you make all of this? Why would you dissipate all of this energy going into unproductive directions, right? That sounds a little bit weird. And one thing that we realized is that if you think about it in terms of what cells actually encounter, they're never going to encounter sort of an infinite 1D line where they always move in one direction, right? What's pretty important probably is that you need to navigate, right? You need to be able to repolarize and go against an obstacle, for instance, if you want to. And for instance, if you think about having polarity alignment alone, and now I'm going to simulate closed channels, so my channels are not sort of infinite, they're closed. If you have only polarity alignment, for instance, your polarity alignment sort of can go in any direction. It's going to go here, and basically it's going to get stuck forever because these cells are perfectly happy like this. They only care about polarity. They don't care about velocity. Their polarities are aligned. They're going to bump against that wall essentially forever. So those are very good at moving very fast, but they have zero efficiency at repolarizing. If you think about velocity alignment now, it might have seemed like a similar term, but it's a bit different because now they're happy. Suddenly velocity is 0. So here their polarities are random walks because they have no information, right? They read their polarity out of the velocity. The velocity of the cluster is 0 because of the things. So it's going to be a random walk where they're going to have to sort of stochastically repolarize. So it's going to take a while. They do it stochastically, right? But after a while, if you wait for very long, they're going to stochastically go back, and then they're happy again because they're going to, as soon as they develop a net velocity of the cluster, they're going to be able to move and they're going to go in this direction. But this is, again, pretty wasteful. So you see, it looked like polarity alignment and velocity alignment were quite different, but if you introduce suddenly obstacles that have obstacles, that distinguishes pretty neatly polarity and velocity, right?
Can just like a free expansion.
Yeah.
That is, can we just have polarity alignment?
I think it's very similar in that case, yes, basically, yeah, that's what I would say. It gets revealed, that's a bit the point of this part of the talk is I think sort of complex boundary conditions sort of reveal things about the interaction of the system that you wouldn't see for an infinite one. And then if you look at now velocity alignment plus contact inhibition of locomotion, it's a little bit better actually because now when it meets this, it's gonna not like it because it's gonna polarize. It's gonna sort of polarize against the boundary. Oh, sorry, I should say. If you have only velocity alignment and contact inhibition of locomotion, you're very good at repolarizing, but you're very non-efficient, right? Because your velocity, you can develop almost a non-zero velocity, and then nothing moves around or moves very, very slowly. If you have the 3 combinations of interactions, you're still quite efficient. So you're still quite efficient at repolarizing. But now, when you meet a neighbor, this one is going to repolarize. And because of polarity alignment, you're going to transmit very fast your polarization. And so therefore, there's going to be like almost an elastic collision where this one, because of contact inhibition of locomotion, this one sort of repolarizes very quickly. It sends that information to the other one by polarity alignment. And so therefore, you spend almost no time against a wall. And you have sort of near elastic collisions for these things, right? So what we were proposing theoretically is that if you now don't optimize for speed in an infinite environment, but if you optimize for speed in a place where you have to repolarize, you have essentially optimal behavior where you have all 3 interactions and they're at near equal contribution, which is very close to the parameter regime that we had extracted. Yes. We looked. It works. It doesn't work as well, and I'm not sure I have a very good explanation for this. Actually, it's we did some. I mean, David did some systematic parameter screening and everything. qualitatively, yes, but it doesn't sort of work as much. And I'm not completely sure I understand why. That's a good question. Yeah. 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 Yeah, it's something that came out of the inference. I'm not completely sure I can understand. I can sort of I can completely explain to you this. This is, I mean, to be fair, I mean they look a bit similar. Yeah, let me think about it. I'm not completely sure. It's a good question. All right. The question is, okay, so that's a completely theoretical interpretation of the data. Is it true that these cell trains are very efficient at repolarizing? That's against a dead end. That's our prediction, right? So the cool thing is that this could be done. This could be done in experiments. They could also micro-pattern channels to stop after a while. And these are clusters. So this is like a relatively thin cluster, and this is a relatively huge and large cluster. But what got us quite excited is that we got exactly the same thing as the data, right? If you look at the trajectory, so these are tracked cells, and this is sort of the density, the probability density of the cluster in time. You can see that you have near elastic collisions, right? Where it's like super fast. You spend almost no time. Bumping against the wall, and you're extremely efficient at repolarizing, and that's also what you see in the data. There's some additional predictions of the model on like the density waves as you collide. There you have density waves, right? Because you start colliding, and there's a bit of an accordion type behavior or something. This is hard to extract from the data. We didn't do it, but I mean, these experiments seem to validate quite nicely the kind of mechanisms that we had. We also played with another model. Yes. Yeah. Yeah, we also looked into this. The problem with that is that it's pretty infrequent, and there was not a lot of data. I mean, there was not a lot of data. It seems to depend on the relative speed. The reason that we didn't explicitly model it is that right now it was quite practical to have like elastic adhesions that don't break, and there we would have to start developing a model for adhesion formation and adhesion breakage, which is, you know, adds a lot of complexity to the model. So that would be interesting to do. But we would have to add sort of parameters on sort of the details of cell adhesion. And I mean, breakage we did, it's not so complicated because you can say that you break at a critical strain, and we do see some breakage sometimes, and that fits very well, and that fits quite well. Adhesion formation is less trivial, right? Because it's presumably requires a critical time. You know, it's a bit more. It's quite a bit more complicated, I think. Yeah.
Your simulation keep the width of the channel the same, but you vary the length of the cluster.
Yeah.
How does it?
It doesn't.
Yeah, but that can't be right because you can't have long range orientation order in 1D short range tracking model. Like the width of your channel will put an exponential cutoff on your code.
Okay, so we didn't. No, you're right. What we didn't look at is we didn't look at. We didn't look at. I mean, so there's two things for this. First of all, technically, it's not short range interactions because you're coupled with velocity, and velocity is a global variable, right? Because if you're elastically coupled, if you're elastically coupled, essentially essentially the velocity is pretty constant, and so velocity is a collective variable of this. So that's one important thing which is sort of interesting. But you're right. We didn't have data for very large. It would be very cool to look at large width and large length. We didn't have a lot of data on this, and yeah, we also small width. It was always only like 10 or the maximum was like 10 or 15 cells or something like this. So we never saw any interesting things. But it's in a simulation. We should do it at least theoretically. You're right. I think it's an interesting idea. We should look at these limits. It's a really interesting point. We didn't do it. We didn't, but I see what you mean. I think it's true that if you have only polarity alignment, I mean, if you have only polarity alignment with velocity, I think you're right. I would expect something like this. You're right. Okay. This is just another small example where it was sort of navigation through complex environments. So it's not having an actual sort of dead end, but it's instead sort of negotiating corners. And it turns out that again, the cells are very efficient at negotiating corners. They only decrease their migration speed for a very, very short amount of time. And that's also consistent with if you compare simulation and experiments, you get sort of quite consistent time scales and magnitude of your velocity reduction. So we think that these mixtures of three interactions that we only parameterized based on sort of moving into sort of infinite space, sort of infinite space, is actually pretty good at reproducing sort of navigation through complex environments. So this is the first part of the first part of the talk. This is the point of it. So this paper just got published last month. It was on Biochemical for a while. And I think the take-home message, right, is that from a more biological side, it seems like it's not just having contacts that matter for your migration efficiency. It's also the geometry of the contact. Having parallel versus perpendicular contacts seems to matter quite a bit. And on a more sort of physics and active matter thing, we think that this is a way that you can extract how a cell interacts with other cells from seeing how they interact with sort of boundaries and physical boundaries, which I think could be a framework. And I think the key thing is that we're looking at sort of intermediate levels, intermediate sort of sizes of clusters, where sort of the difference between the different interactions reveal themselves. Actually, we had started to try to look back at sort of why do certain systems sort of flock in periodic boundary conditions and anti-flock in boundary conditions. We think also we can understand this because, again, periodic boundary conditions sort of allow a net V motion, so that different interactions change depending on whether you forbid or not collective modes. And so I think we didn't do it systematically, but I think you can also understand why periodic versus non-periodic boundary conditions change the mode of migration. Still ongoing. All right. Do I have time? I have a bit of time. Yes. Okay. So the last thing I'm going to talk about is I'm going to sort of shift gear a little bit and go to a slightly different scale. And what I want to talk about here is the fact that actually everything I've discussed so far were purely mechanical model, right? And I've told you that there's been a lot of theories on this, either sort of isotropic active matter, polar active matter, sort of nematic active matter. But one thing that many of these theories have in common to describe these monolayers is that they're purely mechanical theories, right? They don't take into account any internal variables of what goes on inside the cell. And clearly cells are sort of complex biochemical machines that have a lot of internal degrees of freedom, right? And so in a way, what I'm going to add, unfortunately, is an additional degree of complexity to all of these symmetries that I've talked about, which is to try to convince you that you also need to consider sort of internal biochemical degrees of freedom that happens inside the cell to try to understand their collective behaviors. So the thing which I want to talk about here is again this kind of global motion of this, and as I said, this has been described through many different ways. One of the ways that has been described from this, and actually I think Sheila had the original model for this. So the original I think experiment is from Chapitre-Repas, where they found sort of density waves happening in these monolayers, and this was sort of first modeled by actually Sheila in 2015. And this has been found in a number of cell types by different groups, right? And you can see that there are a number of oscillations. What was cool is that we had these collaborators, in which I will acknowledge later, from Kyoto, sort of the group of Tsuyoshi Hashima, who were making exactly the same experiment on MDCK monolayers, but now with friend sensors. And so what friend sensors do is they image the activity of a pathway. And it's very cool because the problem very often is that a lot of these force-generating machineries, what matters is not really the absolute level of a protein, it's about its activity, so it's about its essentially phosphorylation status or something like this. So the traditional thing that you would measure in biology is you would tag a GFP on something to see how much concentration of a molecule you have, but that's not very instructive. What you need is really the activity of the molecule, and what these FRET sensors do is they're really sensitive. They only do fluorescence based on conformational changes of whether the molecule is active or not. And so that's very cool because that can really tell you whether some force-generating machinery is active or not. And so here, these are essentially nuclei. So a cell is here, so it's the same scale. This is like 1,000 cells on a 2D monolayer, on a 2D substrate. And it's color-coded for the activity of this pathway, which I will talk to you about later. And what you can see is that you see this kind of sort of complex waves of everything. But what was really exciting for us is that beyond the motion of cells, you have this beautiful sort of waves of biochemical activity that sort of span through the monolayer, right? Which look very much, which look very much what you would want to do in, you would want to write some reaction diffusion equations to try to interpret this kind of thing, right? And so we wanted to understand, okay, do these things matter, and do they have a relationship with this kind of complex instabilities that we see in vivo? And this is some time ago now. The first paper is some time ago, but I hope to present some more recent things. So this is from my first PhD student, Daniel, and this is all in collaboration with the group of Tsuyoshi Hirashima, who was in Kyoto, now is in the Mechanobiology Institute in Singapore, and this is from his PhD student Naoya, who's now in IST, actually in the group of C.P. Eisenberg. So the first thing that we did is to look at, it was known that there were density waves in these monolayers, and now we have waves of this ERK activity, which is a key pathway for force generation. So the first thing, are they the same? So you can try to do some correlation analysis, so you can look at the intensity of these things, and you can look at the local cell density, and essentially they are the same. So they have these waves have a similar wavelength and period. Yes, sorry. ERK activity, yes. Yes. Sorry? Yes, this was ERK activity, yes. Yeah, it's exactly the movie I showed was an ERK sensor for ERK activity. And so if you look at the chemograph, you can see that there's this. You can see sort of the traveling waves. They have a pretty constant traveling velocity, right? There's also a period in this, and basically they correlate quite well together. And if you do a cross correlation analysis, it's small, but you see that it happens a little bit. It's a bit off-centered, which means that area changes happen a little bit before, are positively correlated with ERK activity changes, and happens a little bit before. Basically, you have first area and something ERK. And then essentially what we wanted to ask is, okay, are those really sort of mechanochemical waves where they really come from these sort of, from the interactions of biochemical activation and mechanics? And so the general idea that we have here, right? ERK activity, some and cell shape, and those cell shape change can be sensed back by a mechanosensation. And so we wanted to write the simplest model you can imagine to take into account this kind of couplings. So the first thing we need to do is we need to develop a model for density, right? Because we are interested. There are density waves in the system, so we need a mechanical model for the density. And for this, I want to. It's a perfect occasion to introduce sort of a 3D extension for the vertex model. So you've heard a lot about the 2D vertex model, but cells are 3D, right? And if you look at the cross-section of a tissue, these cells are not only sort of hexagonal in 2D. So before, the vertex model was describing this red surface here, the top surface here, and it's kind of nicely hexagonal. But if you look at the cells in 3D, they also have this relatively regular sort of nice prismatic shape, right? And so the idea is to do exactly the same, but just to say that the reasons why these cells are very elongated is because they have three tensions: red, green, and blue. And these three tensions have no, because it's so biology, these three tensions have no reason to be the same, right? And if you imagine that the blue and the red tensions are very large compared to the green tension, it's going to be like something that constricts you, and it's going to make a cell very columnar, right? So essentially, what we're going to write for the cell density is an equilibrium-like description based on the 3D vertex model. It should be said that something which is very important in considering 3D is that essentially cells can be described as incompressible, because what defines cellular volume is essentially osmotic effects. Osmotic forces are orders of magnitude larger than cytoskeletal forces. So when you change your tensions, essentially you change areas at constant volume, right? So when you see in 2D a density change happening, it's because the cells are flattening out in 3D, right? The 3D. So what matters really is the aspect ratio of cells rather than the volume, because, and this is something that has been described and measured in quite a number of settings. For instance, in a Drosophila embryo, when the cells invaginate and make some complex 3D shape changes, their volume is really constant. which again makes a lot of sense based on just simple orders of magnitude of osmotic forces. So what do we write? So what we can write is we're going to write the model in 1D for now. And so if you write a cross section of this, again, if you see sort of area changes in 2D, it's going to be linked to the shape to the aspect ratio of these two cells. And this aspect, so you can write just an energy of the system, which is basically just this height here times this tension, and this length here times the tension, right? This is just again just surface tensions times area. I mean, it's just surface tension times length. So that's your energy, and you can write that ERK, your biochemical activity, can change either of these tensions in an arbitrary way, right? I mean, we have no first principles to tell us that. What this tells you is that basically, the aspect ratio, one cell wants to have an aspect ratio, which is a length L0, which is the ratio of these two things right here. So the aspect ratio of the cell is dictated by a ratio of these two tensions, and because these two tensions can be changed by modified by ERK, there's no reason that ERK changes the two in the same way. So basically, what we're going to say is that this rest length here is dependent on ERK in some arbitrary way. Now that's the energy of one cell. Because all cells are bound to each other and are under frictional contact with the substrate, it's very easy. I hope it's easy to convince you that you can coarse grain it into an overdamped chain of springs, right? Because essentially every cell wants to have an equilibrium length. Every cell is there for a spring. It's connected it's connected to springs by others, and it's under frictional contact with the substrate. So that's an overdamped chain of springs. Alone, it's obviously not going to give rise to waves because it's dissipative. But the thing which is nice, which doesn't show up in usual models, is you have a model of an overdamped chain of springs where your rest length now, the rest length of the spring, depends on your biochemical activity is a variable in a system that's going to evolve in time, and that's going to be the key that gives rise to your wave instabilities. And it should be said that, of course, the sign of the coupling, whether ERK increases or decreasing the rest length of the cell is completely arbitrary and needs to be, I mean, we need biological experiment. There's no first principles that can tell us what this is. So what do you write? So what you write on this, the absolute simplest equation you can write based on that is essentially three equations. So the first equation is on the cell position, R. And this should be familiar because this is essentially just an overdamped chain of springs, right? If you write the equation for an overdamped chain of springs, you're going to get a diffusion equation on cell position, where your tau R here is essentially the friction to the substrate. I mean, it's the ratio of the friction to the substrate to the stiffness constant. So this term is very familiar if you just write a chain of spring in frictional contact. There's this weird term here which you're not used to, which is the rest length, right? Usually the rest length is constant, so it drops off from these models. But because here the rest length is not constant, it shows up as essentially a flux, right? It shows up here as a derivative. If different cells have different rest length, they're going to push on each other, and so it's going to create differential movement. So then you need to write an equation for that rest length, and I said it would depend on ERK. So the simplest thing to write is just first order. Dynamics with the delay. So we're saying that basically the rest length depends on ERK with some coefficient alpha and some time scales tau L. It's the absolute simplest thing you can do, right, to linear order. And the other thing we can say, but then we need an equation for ERK. And the simplest thing again we can say is that ERK changes with the delay and follows in a mechanosensitive way the area of a cell. So basically, if R is the cell position, dx of R is the position of the cell, right? If is the length of the cell because it's Ri plus Rx plus dx minus R of x. So it's the derivative. So this is basically a term that tells you that the ERK is mechanosensitive and depends on the area, on the length or the area of a cell. So this is the absolute thing. This is the absolute simplest theory written at linear level. Surprisingly, this is actually sufficient. If you do the linear stability analysis of this, it actually gives you a linear instability at finite wavelength and at finite period, which is nice because it's exactly what you want, right? So and not only that, but it's really simple. It's only the product of these two couplings. So this is the coupling from chemistry to mechanics. This is the coupling from mechanics to chemistry, and it's only the product of the two that matters. So you get an instability if the product of the two couplings is larger than a threshold that depends only on the 3 time scales of the system. Yeah. Yeah. Next slide. Yeah, that's the next. Yes, that's the next slide. Yes, I'm going to show you, I'm going to parameterize everything in the next slide. Yeah. So this is just to tell you something that if the signs have the right coupling, and if this is the model, this gives rise to exactly what we want, right? Because this gives rise to some oscillatory instability at a finite wavelength and period. But the question is, of course, is any of what I wrote true? And can we estimate the parameters? So what Naoya and Tsuyoshi could do is like 2 amazing set of experiments, I think, is that you can test very directly either side of the coupling, because what they can do is they can optogenetically activate ERK, and what you can see is that the monolayer shrinks if you do that. So that tells you that there's a coupling that validates that activating ERK decreases cell area, right? And that's not only that, but that gives you a time scale here in a system, right? It's actually deeper than this. From this single experiment, what Daniel managed to do is constrain all 3 parameters of the. You can get all 3 time scales of the system from this single experiment. Why? Because when you optogenetically activate, you see how quickly ERK goes up, and that gives you the time scale for ERK. You can see how quickly the area of the cell shrinks, which is like a few hours, which gives you the time scale of area changes. But you can also see that the stress propagates. First, this cell sort of shrinks, and then the stress propagates inwards, so the strain propagates inwards, and from the diffusion of mechanical stress, you can get the friction in the system. So actually, it's something really, it's an experiment which is really cool, where you can get all 3 time scales of the system through a single experiment. The other question that we had is, can we do the reverse experiment? Can we test the link from mechanics to chemistry? Well, they can. What they can do is they can sort of put these cells on an elastic substrate, and they can either stretch it or compress it to change the area, the area of the cell mechanically. And what you can see is that when you decrease cell area by compression, you inhibit ERK. So that validates again the other side of the coupling. Not only if it validates the side of the coupling, but actually from this, even without a model, you might see already why there's an oscillation, right? Because that's classical activator-inhibitor dynamics, right? If you activate ERK, you decrease cell area, but if you decrease cell area, you inhibit ERK, right? So that's the simplest, absolute simplest way you can get oscillations in dynamical systems, right?
ERK inhibitor, like once ERK gets activated.
Yeah.
ERK inhibitors also.
So we looked at this. Yeah. So we looked at this. Basically, the question that we have is like, so what they did is that when you compress the cell, you see ERK going up. You can extract the time scale of it. The question that you have is that in the theory, it should stay up forever because you constrict the things. It's not true. It does adapt. But the key thing is that it adapts on time scales of a few hours, which are longer than the period of the oscillation. So we incorporated adaptation in a model, but because the adaptation was happening at longer time scales, it doesn't really change the argument. So we consider that, and it doesn't change the model. Okay. So the cool thing is that from these perturbations, you can essentially extract all relevant parameters of the model, and then you can actually you can get analytical predictions for the wavelength of the instability and its period, which depends again only on the 3 time scales of the problem. But I've actually constrained all 3 time scales, and so I can plug this. And what Daniel found is that he found a period of a wavelength of the instability, which is sort of 10 to 20 cells. This is taking into account the standard error, the error of estimate of all of this. And when you look at the period, it's like 1 to 2 hours. And if you simulate a chemograph, it's basically exactly what you see. It's exactly consistent with the data. So this is like a simulated chemograph with these parameters and this, and basically these things agree quite well. And this is parameter free, right? There's no adjustable parameter in this theory. So that's nice, right? That sort of validates that I think we have a relatively nice explanation for it. And by the way, if you look at these analytics, you can even understand the mechanism for this, right? Because you can see that the period of the oscillation only depends on 2 parameters, which is this activator-inhibitor thing, right? This is how quickly the area changes with respect to ERK, and this is how ERK changes with respect to area. Often with activator-inhibitor dynamics, essentially the period of the oscillation is some kind of geometrical mean of your 2 things. Whereas actually the wavelength depends also on tau r, which is the friction of the monolayer. And that makes a lot of sense if you think about it from the, because you could also think about this as some type of synchronization mechanism, right? Some kind of Kuramoto model. But the key thing is that here, because the phase of your cells are correlated with their area, there's no holes, and it's a finite size game, right? So not everyone can be contracted, and not everyone can be expanded, because again, the total area is constant. So that means that the friction essentially enters this, because what the friction is how many cells can be in phase on essentially a high dynamical, how many cells can be in phase based on the friction of the system during one period of the oscillation. So you can actually interpret quite nicely all of these things analytically. But then the last thing I want to raise in these 15 minutes that I have is I want to ask, and again, maybe it's too anthropomorphic, but this sounds very useless, right? Again, I mean, why do you have like oscillations that go in every direction? It looks a bit pointless, right, to have all of these things. So can we find a reason for these things? Is there a function for these mechanochemical waves in monolayers? And one of the key things that we thought about is that if you make a wound healing experiment, so this is a chemograph of a wound healing experiment. This is space and this is time. So I've shown you before, right? Cells are very good at migrating towards a gap. And if you do the same experiment with ERK sensors, what is very cool is that the direction of the wave breaks symmetry. So you see that the waves start from the wound and sort of propagate sort of essentially infinitely far in the bulk, right? And this was quite cool for us because this is to remind you, this is something I showed you in my first lecture, right? What we know also with these things is that they have long range order, and there was always a question of how do these know that they should go in this direction? Of course, again, you could write down a frocking term, right? You could write down a frocking term where these things are aligned towards the boundary because they see it, and there's a frocking term that tells these cells to migrate up. But it's not very mechanistic, right? I mean, you can write it, but it's a bit sort of ad hoc. And what we wondered is whether these waves were a communication mechanism that could tell the cells in the bulk that they should migrate towards the edge, right? That sounds like a nice communication mechanism to do. So what do we do next? What I should say in the first part of so far in these mechanochemical waves, this was actually a purely isotropic active. This was a purely scalar active matter theory, right? I didn't put any polarity of the cells. I didn't put any nematic. I didn't put anything, right? This was a purely a scalar isotropic theory. And now, because I have like migration, I want to add. Because I have migration, I want to add polar order. The good thing is that because I have a scalar theory, there's very few vectors that I'm going to be able to make, right? To add to my polarity. So I can write now an equation for polarity, which is some Landau-Ginzburg type thing, where these MDCK monolayers are quite different from the. So this fish keratocyte, they moved forever basically when they're alone. So that's why we develop polarity around a finite value. Here, these MDCK monolayer, if you put them alone, they basically sort of go around. They don't have any really clear net polarity. So that's why we actually develop around the state P equals 0 in this thing. It's the only difference from before. We write some kind of diffusion of polarity, sort of again Landau-Ginzburg type equation, and then you can ask, okay, I do need clearly to couple polarity to something, and one of the only vectors I can make based on my existing theory is having, and I'm going to make again everything in 1D, is gradients of stresses, right? Which again is something that has been measured in different cases. So the question is now. First, what I'm going to do is I'm going to sort of forget about the first part of the model. I'm going to, not completely, but I'm going to impose a wave of ERK. So I'm going to make a wave of ERK go towards left to right. And I'm going to ask, that's going to create a wave of stress. And I'm going to ask, is this enough to polarize? If I put a wave in one direction, is it enough to polarize in the reverse direction? Do you think it's going to be enough? Yeah.
Trying to link this to your earlier
yeah.
Where you said that polarity should align with velocity.
Yeah.
Here you are aligning polarity with gradient, correct?
Yes.
That is just velocity.
Yes, exactly. Yes, yeah, exactly. because there's no boundary condition and everything there, it's exactly the point where you were getting into it, is that because your force balance imposes V equals gradient of, I mean, essentially imposes V, I mean, okay, force balance is P plus V equals gradient of P, but your P can be reabsorbed here. So basically just renormalizes your time scale of polarity. That's exactly it. So, so the question is that, so for some reason we were expecting this to work because the wave goes in one direction, so you would polarize in the other direction. This doesn't work at all. And actually, initially we were puzzled by this with Daniel, but actually we shouldn't have, because it's one of the most classical results in the field. It's called the back of the wave paradox, and it's something that people in dictostelium have figured out for a long time, and with many people, sort of Levin, Goldstein, Murray, a lot of people. And the reason for this is that my waves are sinusoidal, right? So I'm imposing sinusoidal waves. So I have a sinusoidal wave of ERK. So my wave of stress is sinusoidal, and I'm only measuring the local gradient. So if you're doing a 1-point measurement of a gradient and your wave is sinusoidal, if I measure here, half of the time I'm going to go left and half of the time I'm going to go right? It doesn't matter that the wave is going in this direction. If you take the derivative of a sinusoidal and you take the average, it's still 0, right? So it's called the back of the wave paradox because it's a bit non-intuitive, but it doesn't work, right? We should have realized this. The way that in the dictostelium people get along this is basically by putting memory in the system. It's basically the idea that is saying that you don't do a point measurement. It means that when the wave reaches you, it desensitizes you. So when the wave reaches the front of the cell sends a message to the back of the cell to desensitize it so that it doesn't go in the reverse direction when it reaches. So it's the classical solution to the back of the wave paradox. In our case, we don't really have a diffusible molecule, but the cool thing is what we have very generically is we have both, because the cells are connected, we have both a chemical and a mechanical wave, and because there's time scales between the two, they're out of phase, right? And so what's cool is that if you have two waves that are out of phase, you can start extracting quite a bit of information out of those, right? And in particular, I hope I can convince you that the maximal information that you have is if you have a pi over 2 phase difference between the waves, right? Because if your waves are in phase, you don't have any additional information. If they're out of phase, we also have no information. If they're pi over 2, it's maximal, right? So that's a bit abstract. Biologically, how could cells actually extract this information? The point is that we need to put a nonlinearity in the system. And the simplest nonlinearity you can say in the system is basically that this coupling, the stress polarity coupling here, this gamma E, depends on ERK, on the value of biochemical activity. Why is this cool? Because effectively what it means is that your direction of polarity is based on your gradient of mechanical stress, but the magnitude of your forces depends on your chemical activity, which means that if you look at the schematic, if you go here, for instance, you're going to go in that direction, but that's going to be amplified by ERK is low, so it's going to be amplified. And here you want to go in the reverse direction, but ERK activity is going to be high, so you're going to kill it. What you can do essentially with the mechanism, without entering into much detail, is that if your ERK activity is out of phase in the correct way, it can kill the part of the gradients you don't like and amplify the parts of the gradients that you do like, right? And so that's a very simple way suddenly that you will be able to break symmetry and sort of go in one direction based on your waves. You can make that a bit more systematic. So Daniel actually could derive an analytical expression. So you can actually measure, you can actually predict analytically the bulk polarization. So essentially the magnetization of your monolayer as a function of the characteristic of my wave. So I have a wave with the frequency omega and with sort of period k. And actually, it has an analytical expression as a function of the two, all parameters being mentioned. And the cool thing is that what this shows is that there's a single optimum of the wave, of a wave period and a wave wavelength that maximizes polarization. And again, it makes sense. It's the one for which there's a pi over 2 phase difference between the two waves. And so you can derive this. That's the optimum. And the cool thing is that the optimum, if you look at where is the optimal of polarization, it's for a wavelength of like 10, 20 cells, it's for a period of a few hours, right? And if you put the data in quite a few systems, it's not exactly optimal, but it's kind of exactly the kind of period and wavelength that you see in a lot of data. By the way, I've shown you in vitro, but there's also this in vivo. If you do a punch biopsy on a mouse and you do a wound in the skin of a mouse, there's going to be sort of radial waves of ERK that go away from the wound. So exactly which with similar wavelength and period than this. So some of these points are in vitro, but some of these points are also actually in vivo, which is cool. So again, it's not a proof, of course, but it's not a proof, and the system is not exactly at optimality. But we thought this was relatively neat still that you can sort of use these waves maybe to polarize against something. The final thing that I'm going to say in the last minute that I have is that in a way this was still a bit artificial because here I imposed the wave, right? All of this was based on imposing a wave, but I had a model from before that made the waves in between before, right? So the next step now is that I'm going to make the full model where I couple my previous model of making waves with my polarity equation. And the only thing I'm going to do is I'm going to have one leader cell at the edge, and I'm going to see if it's enough for bulk polarization. What does it look in the data? So the data, I think, looks quite beautiful if you look at it, if you extend it. What you see is that, so this is time and this is space. What you can see is that initially the waves go in all directions, but there's like a front, basically, where unidirectional propagation of waves wins against bidirectional propagation of wave, right? And there's a nice thing that shows up here. And if you sort of run the full model, if you run the full model, so here I don't impose the waves in any way. All I do is I say that this one cell here is special and is a leader cell and migrates here. You see that there's a feedback between basically where the polarization orients the waves, but then the waves builds up polarization. So this is the wave of ERK. I can also plot now the amount of polarity, and what you can see is that initially the monolayer is polarized, the monolayer is unpolarized and has ERK going in all directions, and there's like a mutual feedback where as the waves become unidirectional, the polarity sort of builds up. And you can see that what this predicts is a gradual buildup of polarity, where polarity starts building up close to the wound and then propagates in the back of the monolayer. And we don't have quite the perfect resolution for this, but if you look at the polarity at two different time scales, initially, so these are experiments. Initially, everything is unpolarized. That's this thing. That's exactly what you expect here. If you look after 21 hours, everything during 1 millimeter is polarized, which is what you see here. And interestingly, if you look at 6 hours at an intermediary time scale, the thing which is close to the wound is polarized, but the thing that is far from the wound is unpolarized, which is exactly what you see if you look in the middle point here, right? So this is quite consistent. So the buildup of polarity accompanies the symmetry breaking of the directionality of the wave, and this makes perfect sense in this model where we couple this sort of isotropic scalar active matter waves to polarity. So basically, that's the time I have. It's again an example where actually there's different parameters. I think there's actually, we think, two instabilities of the system that correspond to two different symmetries of the system. So the existence of the wave themselves don't really depend on the polarity itself. So the generation of the wave is a scalar active matter instability. But then there's a secondary instability when you couple it to polar symmetry, which manages to orient the waves when there's and build bulk polarization when there's a wound. And yeah, that's published. And I won't have time. So this was relatively long ago. This was all 1D, and you might ask, there's some really interesting sort of 2D structure, right? There's like, there's like this propagation. You would want to understand all of this, and sort of what Daniel also did is to generalize the model to 2D by essentially making a 2D vertex model and adding the exact same model of ERK dynamics, and where we try to understand also the 2D patterns of this to try to reconcile essentially seeing this as like mechanochemical waves with previous ideas that they were in the field of seeing this as an active glass, basically, where we combine polarity with mechanochemical waves, and that came out last year. You can have a look. I actually really wanted sort of Daniel to combine this with nematic order to put sort of actually these ERK waves in a nematic field, because I would have really liked to compare systematically. But then he finished his PhD and he defended, so we couldn't get to that. That's something that interests me in the future. And that's it basically. And I'm not going to be able to talk about the fourth problem, which was so the fourth problem I wanted to talk about was actually the open problem, which was here, which was these cells going through disordered environments. But here I'm going to put the slides. I'm going to put the slides on the. I'm basically going to put the slides on your folder so that you can have a look. So that will be. I mean, again, it's something completely ongoing. So we don't have an absolute solution for the open problem. But at least I will put the slides, and that will give you an idea of what we think is going on in the system. So yeah, thank you very much. Yeah.
I also love this.
Yeah. So that's a very good question, and we don't know. So we don't know enough. I mean, so the short answer is I don't know enough about it. I should say so. There's two time scales in the system. What we see is that ERK reacts relatively fast. I mean, if you stretch the monolayer or compress it, ERK goes up and down, up or down in like around 3 or 4 minutes, and that's a pretty. I mean, that's similar to YAP, right? That's a pretty typical time of biochemical activation, shuttling through nuclei from nuclei to cytoplasm. You know, so this is a very reasonable time scale. The time scale that we don't understand right now is if you activate ERK. ERK takes therefore a few minutes, but it actually takes 2 hours for the monolayer to shrink. Actually, so from the thing that we really don't understand is from this activation of a kinase, why does it take 2 hours to compress the monolayer? Because the classical idea in the field is that it's just some kinases that activate myosin, and that shouldn't take 3 hours. I mean, that shouldn't take 1 or 2 hours. Initially, we thought it would be transcriptional, but it's not very clear it's transcriptional. It could be that it's a very, very long chain of reaction that all take a few minutes, and that's why you take a few hours. I'm very confused by this, and the short answer is that we don't know. Yeah. So that's, I mean, that's a slide I removed, but it's a slide I removed because I never have time to read, but I love it. So thank you for raising this point. The question you might ask is, what's my stress polarity coupling, right? And what we think is that Merlin, so Merlin is exactly the molecule that does stress polarity coupling. So Merlin is like a super cool molecule. What Merlin does is a molecule that sits at the cell-cell junctions here, and when the cell is stationary. But when you exert forces on the cell-cell junctions, Merlin goes off of the junction and activates protrusion. So it's exactly what you want from a stress polarity coupling. It's something that gets released from the junction over stress and stimulates motility. So what was really cool, and this is the paper that you refer to. So Merlin is the ideal candidate for stress polarity coupling. And one of the key things in the theory, right, is that the existence of the wave doesn't rely on stress polarity coupling, only its orientation, only it's the symmetry breaking of its orientation. So what Tsuyoshi did is to generate Merlin knockouts in MDCK cells with FRET sensors. And so you can do wound healing experiments in wild type or with a Merlin knockout in cells that have no stress polarity coupling. And I think these things are pretty cool. So these break symmetry and you have waves going in one direction. But these ones not only are bad at doing, so these Merlin knockouts, if I play the movie again, they're not very good at closing wounds. But if you see, their waves are not breaking symmetry. I hope you could appreciate, right, that in the wild types, the waves are all going from front to back. And in the Merlin, there are some waves, but they don't break symmetry. And that's exactly the prediction of the theory, right? In the prediction of the theory, if you remove the stress polarity coupling, you still have waves, but they don't break symmetry upon wound healing, which is the stimulation. So we think that actually Merlin in this system is exactly the stress polarity coupling mechanism. Yeah, thanks. Yeah. Yeah. So yeah. Yeah, so unfortunately, no. Now, actually, so we have a we have a three-way project with both Sylvain Gabriele and Tsuyoshi Hirashima, where now they're putting their cells with ERK sensors on complex boundary conditions. So this is actually exactly ongoing. We have like some shared sort of students that are looking at this, but there's no data yet. But that's exactly we don't have any. I mean, in the trains we don't have any idea of the mechanism, but we're looking at we're looking at ERK now in this context, yes. I'm excited about that. Unfortunately, I don't think so, because these cells are so different. You know, like these cells are so different. I mean, they even the basic thing is that, you know, these NDCK cells compared to the other ones, they don't even, I mean, they don't really migrate alone. I mean, they migrate alone, but they do sort of almost nothing, right? So, and there's honestly, the short answer is I don't know. It's I think it might be very different molecules, but I yeah. All right.
No more questions. Thank you.
Okay, thank you very much.